Welcome back, everyone, and it's a great pleasure now to introduce Professor John Bryant, who is Emeritus Professor at Exeter University in Cell and Molecular Biology. In addition to running an active research group, he has taught biochemistry and molecular biology at all levels, from undergraduate to MSc, and he also introduced one of the first bioethics courses for biology students in a UK university. He's visiting professor in molecular biology at West Virginia State University, where he co-supervises 2 research projects and participates in undergraduate teaching. He's run workshops for biology academics on teaching ethics in the UK, Ireland, Belgium and the USA, past president of the Society for Experimental Biology. He's also past chairman of Christians in Science, and he is a member of the International Society for Science and Religion. Where does it end, John?
Some of that had ended.
Anyway, a man of great distinction. Welcome again. Right.
Well, thank you, Rodney, very much for the invitation to talk in this course. And we say it's a great pleasure to work with Cheryl. This is not the first time that we've appeared on the same bill. It happens from time to time in Exeter when we go to schools together. And Cheryl has also participated in the Bioethics course that I ran for undergraduates from 1995 to 2009. In fact, I had oversight of all the bioethics teaching at graduate and undergraduate level in biosciences for that period, and I'm still involved in teaching bioethics to MSc students in biotechnology and bioinformatics. I need probably to say a little bit about where I'm coming from in this and what my background is. My background is that for most of my research career, starting with my PhD here in Cambridge, working directly or indirectly on plant genes, and for the last substantial chunk of that career, I've been looking at the genes that enable a cell to replicate its genes, which is a kind of interest that we might like to break into at some point. But during the course of that time, back in the 80s, when plant genetic modification techniques were being developed, we had a big grant from the Agricultural Research Council to evaluate one of the possible transformation systems, and my group made a seminal discovery in the way that a particular plant virus works. And then recently working under an EMBO grant with a lab in Switzerland, we made equally seminal discovery in the mechanisms by which the current system actually works, adding one more rather major piece to the jigsaw of our understanding. And so my work has been both academic and applied, and it has involved me in ethics. Actually, I've been interested in ethical issues in relation to science since I was a postgrad, and then moved on to be a postdoc at Norwich, just a few miles down the road. And I gave my first talk on the ethics of bioscience actually as a postdoc back in the early 70s. And so it was almost a natural development of my thinking that I should introduce bioethics as part of my academic portfolio. And I will say that the cells that I've been working on over those years, looking at gene replication, have been the population of cells that are effectively the plant equivalent of stem cells. So genes and embryos then is one of the facets which I've been particularly interested in and have worked on during my interest in bioethics. Oops. So there are ethical challenges. So this isn't going to be deeply scientific, and it's not going to be deeply theological either. It's going to pick up, if you like, where Cheryl left off, and I'll say a tiny bit more about focusing on human embryos. We're going to be looking at IVF, because actually in vitro fertilization provides the practical background to much of what we are now doing with human embryos. I went to go on to think about genetic selection of embryos, including the term saviour siblings, which we will unpack a little bit later, and then think very briefly, and that will be very brief, about direct genetic manipulation, maybe just throwing you out a question or two to think about. And then finally moving on about embryonic stem cells. So all comes under this umbrella of human embryos. Sections of the talk will be split up by a completely relevant picture, but it gives me a chance to show bits of North West Scotland, an area to which I'm emotionally devoted. So it raises these questions that will come up as any thinking scientist starts to ponder on what are we going to do with these data, how should wider society make use of scientific knowledge. It's part of our kind of whole science and society scene. But in particular, are there rights and wrongs about what we can or cannot do with our scientific data? We can, but does that mean we ought to? That was the question posed by the character played by Jeff Goldblum in Jurassic Park, you may remember. He was very, very anxious about resurrecting dinosaurs by effectively a cloning technique. So although I'm very much of the ilk that there are no limits to the sorts of knowledge that are available to us, I think the question we're asking is, how do we apply knowledge? Are there limits to the applications of knowledge? And if so, does a religious faith help us to make those decisions? And this is why it's so nice to follow straight on from what Cheryl was talking about. And I'm a great fan also of Richard Hayes, who does not believe we texts, whether it's the Bible or if we're Islamic, whether it's the Holy Quran or whether it's the Jewish books. I do not believe we can go and find proof text just to sort of pull out and say, that shows us what we should do with genes, that shows us what we should do with embryos. But some of the approaches that Cheryl has outlined, I think we need to espouse, and which particular approach you take to working out what's right and what's wrong will depend very much firstly on which particular religion you espouse, Islam, Judaism, Hinduism, Christianity, and also maybe even the strand of the particular religion that you are devoted to. So, I mean, I would describe my own spirituality as taking a line between Celtic spirituality, meditative, reflective, drawing on the environment, emphasizing the mysteries, and charismatic, emphasizing that the gifts of the Spirit are here for the church today and God is immanent. And I kind of walk the line between those two, and that will affect the way I think about these issues too. So is there a religious view? And that is really part of the question that I'm going to throw out at you. Is there a religious view? Can you use your religious faith to work out what to do in these complex issues? This was part of the Royal Academy of Art summer exhibition in 2002. Mauro Pericetti was very kind and said I could use a picture of his sculpture. And so these are jelly babies embedded in a set resin inside a cross made of a plastic. And the sculpture is called Cloning Religion 2002. And it struck me as a very, very interesting example using the cross in which is embedded the idea of multiplying human beings. So I say Mauro is a very nice man and said, use the picture whenever you want. So I want you to do some work because Cheryl has thrown out some ways that we might think about ethical issues in science. And here is a real example. And as I put up this real example, I want you to unravel what are the actual ethical issues that get sidelined by the wrong ones. Having identified them, how do I decide here what is right and wrong? How did I arrive at that decision? What helped me to make it? So here's the discussion point. We spend a lot of money in the developed countries helping people who are subfertile or infertile to have children. And one cause of infertility is the ability to produce gametes. Now, if the inability lies on the male side, then it's quite easy to get donated sperm, even with the new rules about abolishing anonymity of donors. Sperm donation is a straightforward process. However, egg donation is rather more difficult. And in countries where donors are not paid for their donation, and I know that there are representatives here of some countries where donors are paid for egg donation, countries where donors are not paid, there's a severe shortage of ova. The UK is one such country. Let's go back. And so what happens here in Britain is there's a long waiting list. If you've chosen to have a donated egg as part of the way of treating your infertility, there's a long waiting list. And so we have ovum tourism. For some unknown reason, that strongly Roman Catholic country, Spain, has got an excess of eggs, so people go to Spain. But mostly they go to Eastern Europe, particularly Croatia, Moldova, and places like that, where there is an excess of eggs because people are allowed to be paid for them. So ovum tourism. But if we look at the development of a human during pregnancy, female fetuses by the end of the first trimester have already got nearly a lifetime supply of immature oocytes. And so it has been suggested that because of the large number of abortions that are going on, and it is a very high number in Britain, many of those fetuses are female, why don't we use those fetuses to extract the immature oocytes, mature them in vitro, and then use them for helping women who don't produce their own? So your task in the next 3 or 4 minutes is to talk about that with your neighbour and work out whether you think that's right or wrong. And I will give you 2 or 3 minutes. So I want to hear a great buzz of conversation as the buzz groups form. Okay. Actually, this is worse than any student. So let me say, I'm not going to ask your views at this point, but you can see the sorts of things we're talking about. There's a lot of tangled stuff going on here, and how you apply your ethical thinking is not necessarily immediately obvious. That is a very real problem. The Human Fertilisation Embryology Authority in the UK put that up to public discussion a few years ago, and in the end rejected the idea that this should be done. And I can talk about that in the question time afterwards if you want to. But nevertheless, in countries very near here, there's still active research going on this very possibility. In the Netherlands and in Israel, for example, active research in this area. So that gives you a kind of taste of the sort of difficulties of applying conventional ethics and maybe a religious thinking to the complex issues thrown up by research on human development. So let's move on. As I say, I want to really do a little bit on in vitro fertilization because it forms the kind of platform on which all our understanding of and possibilities of use of embryos depends. And it also illustrates beautifully how, until recently anyway, a lot of research science went on completely unknown to the general public. When I was completing my PhD here in the late 1960s, in the next door building was a crazy physiologist who was trying to get mouse embryos to develop in the lab after in vitro fertilization. He's called Dr. Edwards. We thought he was balmy. It never worked. Surely you need a fallopian tube and a womb and all that sort of thing. But 10 years later, Louise Brown was born because in a collaboration then with Mr. Steptoe in Lancashire, they became the first people to successfully treat a subfertile couple by in vitro fertilization, and baby Louise Brown was born, and the date up there makes me feel very old. And the fact that she's had 2 children of her own also by normal intercourse, not by in vitro stuff. But nobody knew about this. It just went all on in the background. If I hadn't been in the next door building to Dr. Edwards, I wouldn't have known about the research either. But it took quite some time before the government realised that there were kind of ethical issues here. Regulations were needed, and so they asked Dame Mary Warnock to convene a committee to discuss the medical ethical practice that should be applied to in vitro fertilization. And this was part of their report. The early embryo is not yet a person, but nevertheless should not be regarded just a ball of cells. And this kind of raises this whole question of personhood that we were talking about yesterday with Bill. It raises where we place the boundaries of our moral community. Does our moral community encompass the early embryo, or does it not? And that was the Warnock Committee's recommendation, that we won't have a cavalier attitude to this. It wasn't, that anybody can do it and you can do what you like, but nevertheless, we're not going to be regarding the pre-implantation embryo as a person outside the moral community of personhood. I need to say that actually there was a significant minority report that went alongside the Warnock's main report of several thinkers on that committee who disagreed with the main conclusion. Whoops, I keep pressing the wrong button. But it then took another 6 years to enshrine those recommendations in law under the Human Fertilisation and Embryology Act 1990, which has had 2 subsequent iterations, particularly the one in 2008-2009, which made some major revisions. And essentially that way of regarding the human embryo therefore has been enshrined in UK law since 1990. The early human embryo is not a person. And these are pictures taken from the journal The Biochemist, obtained originally from a fertilization clinic, and this is what we're talking about. Here is a newly fertilized egg. These are in an in vitro fertilization clinic. Here is the embryo dividing, going through the cleavage stages. These normally happen, of course, in the woman in the fallopian tube, these stages. finally reaching the blastocyst, which is the stage at which, if that little entity is successful in clinging onto the wall of the womb, and maybe 20 or 30% of them are successful, the woman will become pregnant. So down here she's not pregnant. At this point, a pregnancy could be established if the blastocyst manages to hang on. And we're going to be looking at the blastocyst again later in relation to stem cells. So this is what Warnock was talking about, the early human embryo. And experimentation on the early embryo cannot take place more than a further few days after that bottom right-hand stage. So the rule said that, if you like, implantation is quite a serious event, and the timing of implantation affected, and the beginnings of differentiation, the beginnings of the formation of structure, determined when experimentation should stop. So that then laid out a whole framework of what could and couldn't be done. And then since then, we've had a large number of developments in the technology, making it more efficient, effectively, making it a more efficient way of helping subfertile couples to have children. And I'll just show you one of these. This is ICSI, where an egg is, instead of incubating the egg in a suspension of about 100,000 decent sperm in the dish, one sperm is injected. So the sperm has got direct access to the cytoplasm and then the nucleus of the receiving egg. ICSI, intra-cytoplasmic sperm injection. And a number of varieties of ICSI are available, but when decent sperm are in short supply, or when the sperm themselves are not very active in kind of mounting that attack onto the wall of the egg, then ICSI is often used to ensure that the one sperm gets in, and that's become quite a significant part of in vitro support for subfertile couples. But we've had a number of iterations of the law and changes, and a number of issues have come up. Some of these are, if you like, biomedical issues, and some very specifically relate back to the ethical issues around the embryo. But I just want to talk about these more kind of public ethics issues. In Britain, there's a great debate. In Britain, where we have social medicine, there's a great debate about whether this should be available under the National Health Service. And recently it's been decided it should be. In the past, some area health authorities allowed it and some didn't, so there was postcode lottery in terms of where you lived. But now it's been decreed that all health authorities should offer at least 3 attempts at IVF to enable a subfertile couple to have a baby. But after that, it stops. And if you want to do any more, then you have to pay out of your own pocket several £1,000s per cycle of IVF. And then there are issues about the availability to the couple concerned. How old is the prospective mother? The older the mother, the lesser chance of success, and clinics who are being monitored for success rates are very uninclined to work with women over 40. And indeed, in social medicine, in NHS medicine, NHS clinics will not treat women over 40 with IVF. Private clinics, the guidelines set out by the HFEA, the Human Fertilization and Embryology Authority, suggest that 50 is the absolute upper limit. And although we've had mothers who've given birth in their 60s in Britain, none of those have actually had the treatment in Britain. So they've gone to Belgium, they've gone to Turkey, they've gone to previously Italy, but Italy's now clamped down. They've gone to Canada. So women have conceived and given birth into their 60s, but in Britain we've got quite a clear statement that, I don't know if that's based on natural law or just kind of practicalities, but there is this upper limit. So as I say, in other countries, age, what's that got to do with it? Give me the money and I'll have a go. And then in the recent iteration of the Human Fertilization and Embryology Act that went into law in 2009, up until then, any clinic had to take into consideration the child's need for a father. But under a lot of lobbying from the gay community, that was abolished, and so no longer is a clinic permitted to say, well, this child wouldn't have a father, therefore I'm not have to go ahead with it. So that has been changed, and a number of other, if you like, elements of the social background to IVF have been changed, basically as a reflection of where UK society is moving at the moment in terms of equality and diversity. but that was the one that stood out for me. We're going to be looking at another one of these later. So the law has changed, but nevertheless what we're seeing is that in Britain some 10 to 12,000 babies are born each year by IVF. The technology is well established. We know how to handle human embryos in the clinic. And alongside research on the ubiquitous mouse embryo, that's enabled us to do things with embryos which we wouldn't have thought possible back in the late 70s. Here's one of these irrelevant pictures. I can show you that I'm going to now change tack slightly, and I'm going to move on to thinking about selecting and rejecting embryos. One of the things that obviously the public were not 100% aware of until the Warnock Committee presented its findings was that when you do IVF, you make many embryos. In an average IVF cycle, anything between 6 and 12 embryos are made, depending how many eggs have been superovulated from the woman. And it became into public notice at that point that actually you don't use them all, some are discarded. And so we have now this thing about those look decent under the microscope, I'll use that one to start a pregnancy, those look awful, I won't. And then all across the developed world there are now liquid nitrogen freezers full of embryos that are not being used to establish pregnancies. And so the spare embryo scenario then becomes embedded in our thinking. And it will become even more embedded in our thinking as we move on to think about genetic selection. And I want to start thinking about genetic selection by just thinking a tiny and not a very deep way about heritable diseases. That about 4,500 diseases may be ascribed to a mutation, and what that mutation is might be very complicated, as I'll say in a moment, but to a mutation in a single gene. So you've got this change in the DNA leads directly to this disease. Now, in some cases, it's not 100% penetrance. There's a one-to-one correspondence between the disease and the gene, but you don't necessarily get the disease. So the breast cancer, one mutation, for example, gives about an 80% chance in the lifetime of having breast cancer. The colon cancer mutation gives about a 70% chance in the lifetime of getting colon cancer. But these are nevertheless one-to-one correspondence between the state and the gene. But increasingly we begin to understand that the disease state or the state of health is multi-genic. Genes interacting with each other, genes interacting with the environment, genes and environment acting together. And so we find, for example, that some people are more prone to virus diseases because of a complex interaction of their genes related to the immune system. So that colleague that you thought for years was actually skiving and not turning up for work when they should. Actually, no, they really did have flu again. And their immune system, for reasons of some complex genetic interplay, is not quite as good as yours. So, very subtle susceptibilities. And Francis Collins believes that all disease states of all sorts have got some sort of genetic component. So here's some examples of the direct linkages. Recessive illnesses where in order to have the actual condition you need to inherit a copy from mum and from dad. Cystic fibrosis, very common amongst the Celtic communities, of whom I count myself one. Sickle cell anemia, very common amongst the Afro-Caribbean communities, and therefore in those descended from African slaves in the USA. And then we have the dominant situations. Huntington's disease is, I think, Dennis, you mentioned Huntington's yesterday. Not a classic mutation, it's caused by the insertion of sequences into a gene. And so the gene is not changed in a classic mutational way. But you only need one copy with those too many insertions in it, and you've got the disease. And then there are the X-linked diseases. These are actually recessive, but they are carried on the X chromosome, and so women with their two X chromosomes are fine because the good copy on the other X will override them. Men with their tiny Y chromosome, which, well, it carries very little. It does carry the testis determining factor, which is kind of quite important to its function, but it doesn't have much else, and depending where you come from, it could well have the gene for air guitar, it might have the gene for football, it might have the gene for not asking the way when you're lost. Sort of, that would be a kind of dominant negative effect. But the thing is that if you've got a mutation on your X, you've got problems. And haemophilia, the gene passed on by Queen Victoria to many of her offspring is the classic example. And if you look at the descendants of Queen Victoria, how many of the males died? Many of them before childbearing age because they bled to death. Haemophilia has a very interesting history. It was actually discovered in AD 200, although the rabbi who discovered it didn't know what he'd discovered. It was discovered when a rabbi circumcised a little boy, 8 days old, this classic Jewish rite, and the little boy bled to death after circumcision, and that was rather tragic. And later on, the same couple had another little boy, and he also bled to death. And the rabbi thought, there's something going on here, and said, okay, I'm going to exonerate any other males born to this family from circumcision, despite the fact it is the key rite that admits us into the Jewish faith community. They don't have to be circumcised. And I think I'd better exonerate the cousins as well. And that was very wise. I mean, he had discovered X-linked haemophilia, but he didn't know it. And that's very interesting, seeing that in his writings. Now, one of the things that the Human Genome Project and all the other associated activity in the human genome, and of course we're standing here just a few miles away from the Sanger Centre, which is the British Centre for the Human Genome Project. One of the things that human genetics has enabled us to do is to test DNA directly for the genetic change that causes disease. Whether that genetic change is simply changing the coding, it might be a loss of the on-off switch in the gene, it might be these insertions that Dennis and I have talked about, but it enables you to test direct for the situation. You don't need the person to be in the doctor's office. You just need the DNA. It's like a symptomless method of diagnosis. And so you may have an adult who's turned up with some sort of obscure condition, which might be a late onset condition, and the symptoms might be taken down by the doctor, but the doctor will simply take a bit of DNA and test it. And we can go right the way down back through our lives. Children can be tested, newborn babies can be tested, and indeed are tested for about 5 genetic conditions in the UK. I don't know what the situation is like in the US, but about 5 genetic conditions to help the parents to manage those situations. Not to do a Peter Singer and euthanize the babies, but to help the parents to manage the situation. We can test babies growing in the womb, and in utero testing for Downs has been around for about 40 years, and we can now take that to specific genetic tests. But what I want to talk about is pre-implantation embryos. Those embryos that are made by IVF can be tested before we put that tiny entity back into the womb to establish a pregnancy. We can test the embryos. And this is it happening. Sue Pickering at King's London is probably Britain's ace, extracting individual cells from embryos. It's a bit fuzzy because the embryo is being protected in a buffer solution, and we're looking at it down a bench microscope, and you can see that it's being held by a suction pipette. It's about 0.15 millimetre across the embryo at this point. These are big cells, make no mistake about it, but still it's tiny overall. In more modern versions of this, of which I haven't got a picture, the cell is often cut out with a laser rather than being teased out. It's the same sort of thing. You take one cell out. When the embryo's got about 8 cells, the embryo recovers from that procedure, and because of the power of DNA technology, in fact we can amplify individual bits of DNA sequence in the lab, means that we can test to see whether the mutation we're interested in, whatever change it is we're looking for. So it might be it might be a direct change in one of the bases, it might be the lack of an on-off switch up here, the promoter, it might be an insertion, it might be the complete loss of a gene, it might be a gap in the gene, but all sorts of mutations can be directly tested on one cell. And as I said, the embryo then goes on to behave normally, which is developed normally. And that's simply a feature of the rather interesting way that mammalian embryos behave. That at this particular point of development, a mammalian embryo, whether we're a mouse or an elephant, it's got no front, back, up, down, left or right. None of the cells are yet committed. And it's not, we don't get any commitments until a pregnancy is established. And so that lack of commitment means that the fate of each cell is flexible. Now if we were a fruit fly, this would not be possible. If you were a fruit fly and took one cell out at the 8-cell stage, a chunk of your body would be missing when you turned into a pupa. And there aren't any fruit flies listening to this talk, so I'm aware, so we're okay. So I'm going to tell you a few stories, and these are all genuine stories. In this particular story, this is a British story, and so the names of the participants are not known, because anybody who seeks genetic testing, it's totally anonymous. And only people can go to the newspapers if they want to. But this particular couple didn't. This is one of the very first examples of genetic testing in embryos. So here's the parents, Helen and Peter. They were both carriers of cystic fibrosis. Now, cystic fibrosis, for those of you not familiar with it, is a disease that affects the way you take up salts from your gut and from your lungs. And patients suffer from very sticky secretions in their guts, so they can't feed that brilliantly, and very sticky secretions in their lungs. And often death occurs as a young person, teens through to the 20s, simply because of secondary illnesses that come in through lung diseases. Although with better and better therapy, cystic fibrosis patients are living through to their 30s and 40s and even 50s, like Jackie, for example. But it is a serious disease, and if you've got it, you would know. When we were having our family, starting our family, some friends of ours, both doctors, their very first child had cystic fibrosis, and she sadly died 3 years ago, aged 35. So she'd done quite well. And back in the 70s, often the first thing you knew about cystic fibrosis was when you had a child. You didn't know you were carriers. This particular couple knew that they were likely to be carriers because of their family history. They were tested genetically. They were both carriers. So what did they do? They didn't want to have a child with CF. Remember, each conception has a 1 in 4 chance now of having the illness. They didn't want to take that risk. And so they underwent IVF, and the embryos were tested for cystic fibrosis, and only embryos free of the mutation were used to start a pregnancy. So we're beginning to say that the genetic makeup of an embryo allows us to select or deselect, to use one to start a pregnancy and to discard the other. And indeed, some of the work done, some of the early work on an embryonic stem cells in Britain was done with embryos that had been deselected because they were carrying cystic fibrosis and therefore couldn't be used to start a pregnancy. So that's the very first case in Britain, back in 1989. And as I said, we don't actually know the names of the parents here because they were anonymous. And Matthew was born, and as far as I'm aware, he's grown up healthy. Okay, now here's a story from the States. So we do know the names of the parents here because they allowed the New York Times to make a video of them. So they went public, maybe to raise money to pay for the treatment. Now, here we've got a dominant mutation. So remember, if you've got one copy of the dominant mutation, then you're going to get the illness, or you've got a 70% or 60% chance of getting the illness. So we have this couple from Chicago, and I always have to explain to my British audiences, these are real names. Chad and Colby, this is not something out of Desperate Housewives. You know, this is a real Chicago couple in a lovely middle-class leafy suburb. Chad has the mutation, and his uncle has just died, and you can look at his family pedigree, and you can see it zapping down through the generations. He talks on the video of it devastating his family. So each child has a 50% chance of inheriting. It's late onset, though. The condition is late onset, that Chad is in his 30s and hasn't got the disease, but he's got the gene. Mostly people are dying in their 50s, 60s from it. So late onset condition. And actually in the States, the penetrance of that condition is 60%. Over here, the penetrance is 70%. We think there might be a very slight variation between the two populations in terms of the actual structure of the gene, but you get the picture that whether you're in the UK or the US, the penetrance is not 100%. So late onset condition with 60% actually getting it. And then with the excellent medical care that's available in the States for colon cancer, the cure rate over there, i.e. the rate of getting 5 years of remission, which is regarded as cure, is 90%. It's not as good in the UK, but it's going up. So we have a late onset condition without 100% penetrance for which treatment is possible. So that's interesting, but nevertheless, Chad and Colby decided to undergo IVF to avoid having a child with the colon cancer mutation. And Chloe was born, and she's now about 6. And if you want to look at the video in more detail, just go to nytimes.com and put in embryo screening video into the search box. and that comes up 6 minutes worth, which I've shown to many generations of students, well, a few generations of students, Chloe being only 6. Okay, here's another American story, and this is one that I used to generate a stage play with, which has been performed in universities and schools over much of the west of England. This is baby Adam. Again, it's a real story, and these are a real American couple. Molly Nash, born with Fanconi anemia. She comes from the state of Colorado, but the treatment we're going to be talking about wasn't available in that state, and so they went to one of the northern Midwestern states. I think it was Minnesota. Fanconi anemia is a genetic disease. It's inherited. It is recessive, but that's irrelevant to this story. It usually causes death between the ages of 7 and 15, that sort of time. And Molly is 7. But actually, it's one of those diseases that can be cured by stem cell donation, a bone marrow donation, if you like, from a disease-free, and here's the rub, compatible donor. And so Molly's parents, Linda and Jack, elected to do IVF and PGD. PGD, if I've not said it before, is pre-implantation genetic diagnosis. I keep throwing the letters at you. Pre-implantation genetic diagnosis. They generated 14 embryos through IVF, of which several were free of the mutation, but only one of those free of the mutation was immunologically compatible with Molly. And I will say that prior to undergoing this, they'd searched the close relatives. Molly was an only child, but none of the cousins and none of the parents were close enough. But one of the embryos was an exact match immunologically to Molly, which means that if the new baby is a stem cell donor, then Molly is not going to reject the stem cell graft. So Adam was born. And again, we're fortunate here because the umbilical cord contains cells which behave like stem cells. They are effectively stem cells. And fortunately, in this particular instance, the umbilical cord provided enough stem cells to cure Molly. And I will say straight away that had they not been enough, then baby Adam would have been subject to a bone marrow aspiration, drawing bone marrow cells out of the long bones of his thigh at some point in the next few months, but they were used to cure Molly. I was reading about Molly just a few months ago. Molly is now a kind of typical teenager, as her mum said. She's calling her friends and what called over their cell phones. We insist on calling them mobiles, and hanging around the shopping mall on a Saturday. I suppose that's harmless. But anyway, normal teenager. She has diabetes. Life has not dealt her a good hand. She's got juvenile onset diabetes, but it's got nothing to do with the Fanconi anemia, but she's coping with that too. However, I mean, that's very positive, but if you want to read about the downside, Jodi Picoult's novel, My Sister's Keeper, set a few years in the future, gives you another story. And I think before we get very enthusiastic about these saviour siblings, because this is what baby Adam is, he's a saviour sibling, then I think we need to think that there could be possible downsides. And I'm going to give you a few data in a moment related to numbers based in the UK. Okay, we're going back to the UK now, to another couple. This is Tom and Paula, and another Molly. Tom and Paula are both theatricals. One's a director and one's an actor. And I can't remember which way around they are. But Tom's given name is actually Tomato. So I imagine he had kind of hippie parents. I use Tom here because if you're talking to school students at this point, they begin to wonder whether you're being serious. So Tom, Paula and Molly. Tom and Paula are deaf. And in both cases, their deafness is inherited. It's not the same mutation. Deafness is very complicated genetically, and very many different causes. But it so happens that they both have an heritable form of deafness, but almost certainly different genes. Molly was born deaf, and Tom and Paula decided they wanted to make sure that their other child was born deaf. And so they applied to their local health authority for genetic testing. Genetic testing of embryos is available under the NHS provisions for serious genetic conditions. But the local health authority said, no, we're not going to select to have a deaf child, we can't do that. So they appealed to higher authority, to the Human Fertilisation and Embryology Authority, and the HFA also turned them down, and subsequent appeals have also failed. And this allows me to mention another very specific provision that's been written in to the new version of the HFE Act, that selection shall not be carried out positively for conditions that are regarded as disabilities. And so the HFEA rejects this application on those grounds. The deaf community are up in arms about this because they say that they have their own language, British Sign Language in our case, and that being born into the deaf community as a deaf person is not a disability. There's been a lot of tension and discussion in the press as to whether this should or should not be allowed. But so far, they've not been allowed to undergo IVF and genetic testing. They are obviously normally fertile because Molly was born through natural intercourse. And the last I heard, at the end of 2009, was they were now trying for a second child by the normal fund method rather than going via IVF. And that's an interesting case, and that's something that you might find quite hard to think about ethically. When I put this to school students, they are all very anti this, including one student who was profoundly deaf. It's very anti the idea of selecting for a deaf child through pre-implantation genetic diagnosis. So they're just 4 case studies of the sorts of things we can do with genetic testing of embryos prior to implanting them, really based on our vast experience of handling embryos in the clinic because of IVF, and the combination of that with the power of human genetics. And so we need to think, what are the key ethical issues about genetic testing of embryos? And I'm not going to tell you what they are. I mean, my job is just, I think, today, is to alert you to what's going on and to get you to reflect on the situation. So, how prevalent are these things? How prevalent are they? Well, this genetests.org is a website run by the National Institutes of Health in the USA, and that keeps a running clock of the genetic tests available clinically anywhere in the world. And as of earlier in the week, available anywhere in the world in clinical use are 1,800 or so. Now, some of these tests are not very informative. Some, if you like, just give you a percentage risk. Some are very, very clearly informative. Those that are not very informative are sometimes not very well supported by proper genetic counselling, particularly if it's a commercial company that set up a testing regime. So we need to be aware that it's not everything is as clear-cut as in the cases that I've been talking about. Now, of that 1849, I don't want you to give the impression that people are testing embryos right across the world in all these cases. In Britain, every new gene that people want to use to test on embryos has to be approved by the Human Fertilisation and Embryology Authority, and they have so far licensed about 100 that vary in seriousness, I would say. So that's another kind of ethical issue. How serious does the condition have to be before we allow it to be used in this way? And I'll also say that although there's a list on the Human Fertilisation and Embryology Authority website, it does not contain all of the ones that are licensed for this reason, that some of the conditions, 2 or 3 of the conditions are so rare that if the genetic mutation was mentioned, it would identify the families. We're very keen on families preserving their anonymity if they wish to do so. So, on the grounds of anonymity, 2 or 3 of the conditions are not mentioned, but about 100 are mentioned on that website. And then to put those figures in perspective in terms of what I've been saying about IVF, as I say, in Britain between 10 and 12,000 IVF babies are born a year. It's probably knocking on the 12,000 more than the 10,000 in the last 5 years. Probably 200 or 300 of those have been subject to pre-implantation genetic diagnosis, that sort of order, 200 to 300. And probably of that 200 to 300, probably a tiny handful, just a handful probably, have been in the saviour sibling scenario. It may well be less than a handful in a given year in Britain. So Britain, including Northern Ireland, with a population of 68 million, 12,000 IVF babies, 200 to 300 have been born after pre-implantation diagnosis, and somewhere between 2 and 5 have been born as saviour siblings. And the HFEA is very keen, when they're looking at a saviour sibling application, very keen that a complete and proper search has been done through other systems for donors, through the Anthony Nolan Trust in the UK, for example, before they will allow the saviour sibling scenario to be enacted. I just want to spend a couple of minutes thinking about eugenics. Eugenics means good breeding or well born. It doesn't mean to say how you hold a knife and fork and whether you crook your finger and hold a teacup or anything. It's talking about being born with good genes. And Darwin wondered, but Darwin wasn't actually a eugenicist, but he did wonder whether we could apply evolutionary thinking to human society, but actually it'd been predated by over 2,000 years because Aristotle and Plato both reckoned it was a good idea to encourage the better citizens in the Republic to breed. And indeed, I think it was Plato that suggested that the better men should have intercourse with the better women as often as possible. But of course, who decides what's better was vested in the liberal middle classes, as it always is. So deciding who's better was another issue. But Francis Galton, who was Darwin's cousin, actually formalised eugenics in the UK and wanted to encourage the fitter to breed more extensively and discourage what he called the underclasses from breeding. And although we think of Nazi Germany as being the prime movers of eugenics, remember that the eugenic theory was first written down in a formal way in Britain at the beginning of the 20th century. And eugenics movements were popular in many, many Western countries through the early years of the 20th century. And when I was a student there, you could still find posters dating back to the 1930s telling you to only broadcast the best seed. And this wasn't barley they were talking about, even though it was a picture of an old biblical sower, sowing the seed. And so in all these countries, the eugenic movements, and in some of these countries, they actually enshrined eugenics in law, including the USA. Negative eugenics, sterilisation of the less fit, the morally feeble. And some 50,000 eugenic sterilisations were carried out in the US up until the 1950s. And the most recent case of eugenic sterilisation that I've been able to find was in 1974 in Sweden. And again, we can discuss why they might have done it. But one question to ponder is, are we now doing eugenics one by one, by selecting embryos with particular genes, improving human society in a different sort of way? Just a question to ponder. And then the other questions are raised is, how far should we go in selecting our genetic makeup? Where are the boundaries in terms of the seriousness of a genetic condition before we say, I don't want to have a baby with that? One of the tests that is applied to newborns is for a condition called phenylketonuria, which is a condition which means that you can't metabolize one of the essential amino acids in proteins. And babies have been tested biochemically for that in Britain for at least 40 years, but they're now tested genetically for it. And you control your diet. The diet becomes rather dull, but other than the dull diet, it's a fairly normal life. But would you include that as a genetic test to deselect an embryo? Just a question. Where are the boundaries? What about non-medical situations? We know that genes have some input into our behaviour because genes affect the biochemistry and the morphology and the physiology of the brain. We know they have some input into our intelligence, although environment is hugely influential in both behaviour and intelligence. What about sporting ability? I'm always keen to see an England cricket team that will actually dominate the world. They can't seem to even dominate Bangladesh. Genes that affect our appearance. The atheist philosopher John Harris, who has been a member of the Human Fertilisation Embryology Authority Advisory Panel, the atheist philosopher John Harris, and his words imply that he was a Van Morrison fan, as you'll hear in a moment. He says that if a couple wants a bouncy, curly-haired, brown-eyed girl, there's the Van Morrison man, why would you not let them choose, rather than allow nature, and then he writes, or their God, to do it for them? And why don't you let them choose? That's what they want. After all, he says, we pay for music lessons, sports training, ballet classes, independent schools. So why don't we just allow them to choose what their child looks like as well? So that view is out there, and certainly there is a medical geneticist in San Francisco who's working very much alongside the genetic research teams, who tells us that selection for eye colour will be about 80% certain within the next 3 or 4 years. And at 80% certainty is going to offer it, selection for eye colour. So just a thought. Okay, and the final bit of genetics here is what about genetic selection? I put the picture of Dolly up there because Dolly was cloned from a sheep that had been genetically modified. What about directly modifying humans? The point about cloning of Dolly was that she was very valuable. And rather than undergoing the whole cycle of GM again, you could clone that GM sheep and you might have a little herd of them. But would you want to genetically modify a future human? I add a gene to that human. And you might want to think the sorts of genes you might want to add. So that's hanging about in the background. It's 100% feasible to do so. Not with a high success rate, but 100% feasible to do so. Okay, here's another irrelevant picture. It's not relevant to me. But shows that I'm going to change cat very slightly for the last few minutes, and we're going to think about stem cells. Because again, it relates to human embryos. Stem cells, the source of many different cell types, and the idea is that we might use stem cells to repair people who have been worn out or damaged in some way. Maybe it's old age, maybe it's trauma, maybe it's disease, but we might want to mend them. And the blastocyst, that little structure I showed you, and here it is again, that little structure there is a source of stem cells because that part of the blastocyst may consist of 30 to 60 cells, will grow into the embryo proper after pregnancy is established, will grow into the fetus, will turn into the baby. By definition, therefore, that is genetically able to encode every single cell type that occurs in the adult human body. So maybe we can use that, culture those in the lab, and in some way induce them to grow into particular types of cell. This is the pattern here. There's the blastocyst cells, we culture them, and we either get them to grow into particular cell types in the lab, or we inject them into the human body, where the niche that we use for the injection, whether it's the heart, whether it's the bone marrow, or whatever, but the niche we use induces them to come along particular lines of development. So you need embryos. You obviously don't allow the embryos to develop any further than that 7-day stage, the blastocyst. We culture the cells and then use them as required. That is the theory. And in Britain, the research is very active, and indeed in 2001, an amendment was made to the then Act to allow stem cell research to be part of all research on human embryos. It was brought under the umbrella. Very much a political decision, incidentally, here, believing that Britain had a kind of European lead in this area. And again, this has to be regulated by the HFEA, the Human Fertilization and Embryology Authority. And so we can have embryos that have been produced by IVF, and all those spare embryos I've been talking about have been part of the kind of scenario that's led to stem cell research in this country, now in several different labs. But the problem with stem cells made from any old embryo has already been raised in connection with Adam and Molly Nash, Adam Nash the Saviour sibling, because if they've been made from any old embryo, you have only a very small chance of those stem cells being compatible with anybody who wants to receive them. So why not make an embryo? If I need stem cells, why not make an embryo from me by cloning? Not to make a mini-me, but to make an embryonic version of me, which can be used as a source of stem cells. It's called therapeutic cloning. Although President George W. Bush effectively banned federal funding from supporting this work, there are many, many biotech companies in the US that are working very hard on this to produce cell lines from individual people that might be used for repairing them. In Britain, the success rate has been very low. That two labs in Britain have been granted licenses to do therapeutic cloning, but so far the success rate has been very poor. Cloned embryos have been produced in Newcastle upon Tyne, up in the north of England, and very controversial is the wrong word, but very famously in Seoul in South Korea, a number of blastocysts were obtained by cloning, but the claims then made for what had been done with those blastocysts were completely falsified. And the very prestigious journal Science was fooled into publishing 2 papers which suggested that the South Koreans were on the verge of actually developing treatments based on cloned embryonic stem cells. They weren't. All that the group had managed to do was to actually produce about 7 human blastocysts, none of which survived long enough to obtain the stem cells from. So that technology is still ticking over in Britain, ticking over rather faster now in the States than it had been, and one wonders about its future. And then finally, the modification of the Act in 2009 gave British scientists the permission to use hybrids. Now, I need to explain what a hybrid was. I think Cheryl mentioned them in passing. But a hybrid is where you use an empty egg of another mammal to be the receiver for your clone donation. So if you think about making a clone, you need an egg, and you take its genetic material out, and you put in the genetic material from another cell that's already got the full 2 copies, and that's cloning. But obviously you need eggs, and I've already said that eggs are in short supply, even for fertility treatment. And so scientists put forward the idea that you might use the empty egg of another mammal as a kind of carrier for the human genetic material that you put in to do the cloning. Not for therapy, but for research. And so British scientists were given permission in 2009. They were already doing it, I need to say, but were given permission in 2009 to use the empty eggs of cows and rabbits to clone up the genetic material from humans. And of course that raises all sorts of issues again about personhood, which you might want to discuss later. And this raises the question, which Cheryl and I have discussed with Ted Peters. Does our motivational mandate to heal? That's part of our, those of us who are Christian, Jewish or Islamic, it's part of our mandate as religious people to want to, if you like, make people whole. Does it allow us to use embryos? And what tools and principles help us in our decision making? And I would just give you an example of 2 ends of the spectrum, both using the neighbour argument. An organisation in Britain called CARE takes a very conservative view of this, and my friend Philippa Taylor, who writes on these issues, talks about how the weakest of our neighbours is the pre-implantation embryo, and therefore we need to look after it. Ted Peters talks about our neighbours as those who are sick and they need repair, they need stem cell technology, and so we use stem cells to repair those neighbours. And it will be very interesting to get Philippa and Ted in the same room. I'd like to do that one day. Okay, now I'm just going to move on very briefly to show actually the scene for stem cells has been changed quite dramatically by some discoveries made over the last year or so. In the winter 2007-2008, 2 groups, one in California and one in Japan, showed that you could take the stem cells which underlie our skin and repair our skin all the time, could be actually induced to behave by a little genetic trick, induced to behave as if they were stem cells. I will say again, I want to bring in a bit of Celtic culture here. It requires manipulating 4 genes, and one of them has a Gaelic name. One of them is called nanog, which is Gaelic for of youth. So the 4 genes that keep stem cells in a stem cell state retain their youthful appearance and their youthful activity. Those are needed to be activated to get our skin stem cells to behave as if they were embryo stem cells, but it can be done. And just a few months ago, it was demonstrated that we could now get them to develop in specific ways. And experimental therapy was announced just on Monday this week. These cells are now being used to see whether particular regenerative diseases can be repaired using these induced pluripotent stem cells. Induced because we have to do a trick to get them to behave like stem cells. Pluripotent, capable of many, many different formations, stem cells. And I think that's changed the ethical climate somewhat. Although the research that was needed to understand embryonic stem cells, and so therefore they took their role, because that's how we understood about the 4 genes, nevertheless, we're now using, you'll see the possibility of using, patient by patient, induced pluripotent stem cells from the patient's own skin. And that's a sort of very, very interesting way of doing therapeutic cloning. And so I guess, you know, we ask the question, you know, will that be enough? Will that sort of research, or research in other forms of stem cells, be enough to eliminate the use of embryonic stem cells? Does the ethical climate become a lot easier? And certainly those vigorous opponents of ES technology, I think, would hope so. And again, I think those of us who've been involved in the debate in Parliament and in public have certainly detected a change of mood in the biomedical community, thinking that they're no longer treading on ethical thin ice. I will say this has been, if you like, a classic example of public versus private morality, because it was mentioned earlier that probably 20% of the UK population are opposed to stem cell research, 80% are for it, and in the utilitarian way that our country's governed, that's okay. And so we go back to those questions. And does the religious faith help us to make ethical decisions in these areas? What form does that help take? Going back to Cheryl, you perhaps realise that we did talk about our joint presentation, as it were, before we came. And if you like, on all the issues, every single issue I've talked about today, you can find Christians who disagree with other Christians, you can find Muslims who disagree with other Muslims. In Judaism, there's actually more of a consensus. Most of it is regarded as okay, except by the ultra-orthodox. Most religious Jews of more liberal persuasion are okay about nearly everything I mentioned today. But certainly there's strong disagreements within the Christian and Muslim faiths. Okay, I'll finish with just a few resources. These are nearly all written by me. But this one, Ethics in Brief, published by the Kobe Lang Institute of Christian Ethics, which is at Tyndall House, just the other side of Cambridge, is where I just review, based around the 2009 iteration of the Human Fertilization and Embryology, I review a number of different Christian views in these areas. And I was hoping we could get some printed copies of that here. But, I don't know how we access copies from
we ought to be able to do that. We've certainly got copies of the other things.
And also in the well-known Test of Faith resources, there's quite a lot of discussion about bioethics. So thank you, Rodney.
Brilliant.
And everybody else.
I'd be really surprised if that hadn't raised lots of issues. Yes.
Sometimes I think the horse has bolted. If we accept ethical boundaries established within a scriptural text, then that actually allows us to think about the question differently. So I think the example you gave of finding different types of stem cells is a perfect example. And sometimes I think it just might, there's a need to accept the ethical boundaries and then think about the questions differently or come up with other solutions. And I think that's worth putting forward. That just because we can do it now doesn't mean we won't be able to do it in 5 years. And maybe it's worth waiting 5 years, because when you do this now and you let the horse bolt, now you have to deal with all kinds of other things.
Yeah, I would like to think that the groups in Japan and in San Francisco who developed the induced pluripotent stem cell lines were concerned about the ethical debate, but I'm not sure that was so. I think they were much more concerned about the practicalities of treating individual patients with stem cells and the difficulties of human cloning. Humans are hard to clone. Whereas what they've done is they've identified in each human being a source of stem cells that can be used to treat that human being without the need for human cloning. And it's very interesting, when the leader of the San Francisco group was interviewed on the BBC, the transcript of his interview, he was actually asked about the ethical side. He said, that didn't really concern me. And he said, I was concerned to get an effective treatment, patient by patient. And he said, if there is an ethical bonus, that's okay. That wasn't the motivation for us doing the work.
Yeah, there's been a fair amount of disagreement, I think, about the notion of one gene, one disease. I was really struck by your figure of 4,500. So I'm wondering, why, could you say a little more about what constitutes the notion of a genetic disease? I'm thinking of your colleague, Lenny Moss, who was celebrated at one point for a book, in a sense, criticizing the notion of a single gene related to a single disease, except in a relatively few cases.
Yeah, well, I disagree with Lenny. Lenny, quite rightly, I think, is a critic of genetic essentialism and reductionism, and I think I'm with him. But nevertheless, there are significant numbers of cases where we can say that if you carry this mutation, this disease will be the result. It may well be that you can ameliorate or modify or even exacerbate the effects of the gene because of interaction with other genes or with interaction with environmental factors. But there is no doubt about it. You know, if you have 2 copies of the cystic fibrosis gene, you will have cystic fibrosis. If you've got one copy of, if you as a male have got 1 copy of the hemophilia mutation, you will have hemophilia. These are clear one-to-one. And my 4,500 are cases where we, despite the possibility of modifying somewhat, where we know that this gene will end up with you having this condition.
Do you mean that in the case of 4,500, there would be straightforward inheritance down and
Yeah, straightforward Mendelian inheritance.
And with 100% likelihood?
No, the penetrance of these conditions does differ, as I said. There is a direct linkage between the gene and the illness, but the penetrance, that is, the actual possibility of you having the illness, varies between about 100% at one end, cystic fibrosis, haemophilia, and about 50% at the other. But nevertheless, there's still a direct link.
Thank you. Yes.
Can you shed some light on the stem cells research and the differences between the embryonic stem cells and adult stem cells? Because I think there's a better success rate when it comes to adult stem cell research than embryonic one. And if so, why is there such an emphasis on embryonic stem cells?
Okay, the emphasis, let's start with the last bit of the question. The emphasis on embryonic stem cells, if you like, comes from an intuition based on our understanding of mammalian development, that those stem cells in nature will give rise in development to all 220 types of cell we find in the adult body. They've got that potential, and that's what they're for. And so therefore we assume that we could tap into that potential and thereby think about repair of any bit of the human body. Now, the adult stem cells, which we all possess post-embryonically, are located in specific parts of the body and relate to the repair or replacement or turnover of particular types of cells. So the cells in our bone marrow give rise almost exclusively, not 100%, almost exclusively to blood cells. They've got 1 or 2 other jobs as well. So, if you like, they have a limited potential. Their potential in nature is to make far fewer cells. The cells underlying our skin make the layers of our skin. The cells that repair liver, if it's not damaged too badly, repair liver cells. And so intuitively, you expect it to be a rather limited range of applications you might have from any particular application of human stem cell, of adult stem cells. But it's been discovered, and there were many, many false starts, and there were many, many data that were misinterpreted. But it has been discovered that adult stem cells, at least some populations of adult stem cells, will undergo what we call transdifferentiation. Will choose they've already chosen one career pathway, we get them to move to another one. And so there have been quite a few experimental therapies with adult stem cells repairing bits of tissue you wouldn't think they were capable of, particularly bone marrow stem cells have been now used in experimental therapies to repair 2 or 3 different types of organ. They haven't yet entered clinical practice, but it is absolutely true that we have not yet had a single clinical trial with an embryonic stem cell line, but we are undergoing clinical trials in the States, in Britain, in Germany, with adult stem cell lines. But the emphasis on embryonic stem cells came from, if you like, biological intuition. This is what they do, so we'll tap into that.
Thank you. Yes.
It seemed to me that in all the cases you have presented to us, it was a matter of chance and not of certainty that the baby will have a specific disease. Can you comment on this a bit? And if you think that whether a Christian or a believer can use this like an argument for, let's say, let us God to decide what's going to happen, and we believe that God has a plan with us, and this is exactly the territory for him to decide?
The term chance here relates, if you like, to a statistical likelihood.
Mm-hmm.
That's what I prefer to say. So that in the case of two parents, each with the same recessive mutation, the statistical likelihood for each child is 25% of them actually having the full-blown condition. Now is that what you mean by chance?
Mm-hmm.
And for a dominant mutation, it's a 50% chance, 50% likelihood. Now, I don't know how familiar you are with some of the genetic diseases and what sort of life a small child would lead if they were born with such conditions. Some of them would lead to a life which is very short and brutish. And the question then is, I think up to each individual set of parents to decide whether or not they're prepared, and it is a risk, with a 50% chance of the child having this to die within a few weeks, whether that's something that they're prepared to do. And I'm not going to speak for the conscience of any prospective parent about whether or not they want to leave that, inverted commas, to God. But that will enter into the equation, prayer will enter into the equation. Richard Hayes is actually very good at evaluating this sort of dilemma in terms of... but the other thing I would say is that, I'm very much a communitarian in terms of my understanding of Christian ethics. And whatever decision that couple comes to, if they're Christians, the church supports them, right? It doesn't say, oh, God, how on earth could you go through that? Oh, you're not part of our community anymore. The church supports them because they prayed it through.
I think that's probably, that's because it's a general principle, whether we agree or not in the first place.
Absolutely.
We've done with people.
I think Jenny's been out long to ask a question.
Yes.
Well, I guess my question or comment just kind of follows on that, because you focused on the embryonic ethical issues, but...
That's what I was asked to do.
Right, good, and I'm glad, but also in your list of genetic testing, you mentioned babies growing in the womb, and I just noticed when I was a child growing up in school, there were other school children with me who had Down syndrome.
Yes.
Where are they now? I mean, I'm just frightened. I look around and I see people being selected out from even getting the chance to be alive because of they're being aborted, or in some countries, many of the female children are being aborted based on these tests. I mean, I'm horrified by this, and I just wondered if you had any comment. I mean, to me, this is much more problematic than perhaps doing the kind of IVF type of ethical decisions, which are also difficult, but just, how do you have any, why aren't we as a society horrified by this? We're sort of deciding who's worthy of living and who's not, and are you being compassionate to allow somebody who's going to have a disease or might be poor even in some cases to are you being compassionate by not allowing them to live? I mean, this is a horrible thought to me. This is the early documents from Adolf Hitler had that same kind of language, right? That we are going to be compassionate by sparing these people of a miserable life by taking it away from them. So just, why aren't we horrified by this?
Well, yeah, I was horrified to hear an American doctor, Richard Seed of Chicago, saying, talking about supporting people with housing projects. They don't do that. He said, you want to eliminate the wrong genes. So it's not just Adolf Hitler. Let's go back to the actual question, though. That before this pre-implantation diagnosis was available, it's certainly true that many genetic tests were being applied to the in utero. And as you rightly say, the Downs test had been available for a long time. But I know many Christian couples who reject having a Downs test because they want to bring that child to term, look after it and care for it. But the number of Downs children being born certainly has been reduced because of the availability of the test. When genetic tests were first available, they weren't available for embryos. They were available only in utero. And there's a very famous case of Professor Tom Shakespeare, who's the professor of medical ethics at Newcastle University, and he has achondroplasia, which is the dominant mutation that causes short stature and enlarged skull bones and so on. It doesn't cause any intellectual impairment whatsoever. But in the past, there's been a lot of prejudice against people who are dwarf, and quite often they could only get employment in circuses and so on, despite the fact that... but Tom is a professor of medical genetics, and when he and his wife were carrying their first baby, the scans, which were coming into regular use in the mid-80s showed that the baby was achondroplasic, and the genetic test confirmed it. And the clinicians here in Cambridge said, well, I think you should terminate the pregnancy. Of course he didn't. And there's a lovely, lovely film that was made by Channel 4 that I've got, unfortunately on VHS, not on DVD, called Ivy's Genes, which is all about his little girl Ivy. But it seemed to me at that point that we were beginning to apply genetic testing to conditions with which people were 100% happy to live with. And who am I to talk about somebody else's quality of life? Which 100% manageable. And so we begin to kind of look for that elusive perfection. But in terms of that versus PGD, if it is a very serious genetic condition, the sort of condition I was alluding to in answer to your question, I think there are certainly couples who would not at all be happy with an abortion because they know that a life is growing in the womb already, but nevertheless feel okay about pre-implantation genetic diagnosis. And I used to say that in theory, but I now know a couple who are in exactly this position. They abhor abortions, but nevertheless they're quite happy to go through PGD to avoid their child having this serious genetic condition, because many people don't regard the early embryo as being part of the moral community in the same way as a growing fetus or a born baby is. So the advent of genetic testing of the embryo has helped some people.
Thank you. Well, now there are...
Wow!
Yes.
Forest alarms
So, yes, a forest alarm.
Thank you for the talk. That was really fascinating. I want to ask about, earlier we talked about evolutionary biology, in evolutionary biology talks. The diversity, by the time diversity is increasing, can we talk about the evolution of diseases? Can we say that the diversity of diseases are increasing day by day as we find more treatment, we solve the problems by time?
It's certainly true that, if you like, the human population has received into, if you like, the human population has received new diseases in my lifetime. And so, for example, HIV/AIDS was probably not a human disease in the 19th century, and probably entered the human genome sometime. Well, we say 1950s, and I certainly was around then, but it might have actually been earlier than that. Some of the viruses, the viruses that cause complete breakdown of the vascular system, things like Ebola, seem to have changed their hosts, maybe in relation to environmental change and so on. So, if you like, the diversity of diseases to which humans are subject seems to be increasing. And also some of the diseases to which we're subject, particularly the viruses, and to some extent, to a lesser extent the bacteria, are very prone to rapid mutation. So we get new strains of influenza and so on. So there is a sense in which diseases are evolving, and some of those are evolving in response to what we're doing to try to avoid them. Bacteria that become resistant to antibiotics, for example. So yes and no, I think, is my answer to your question.
Fine.
Sorry,
Do you want to quickly follow that now?
Now the main diseases are cancers and genetic diseases.
Well, they're the main diseases of the developed West.
West. Okay.
You know, I mean, when Bill Clinton, when the first draft of the Human Genome Project was announced, Bill and Tony Blair had this transatlantic love-in to celebrate the genome. But Bill said, you know, he looked out at this array of students and said, you know, many of you are going to live to 100 because of these developments. And I wanted to yell down the phone, Bill, go to Africa. You know, life expectancy in Zimbabwe was at that stage 38. Millions of children are dying in Africa because of diseases that are completely preventable. And so although the diseases of degeneration and of cell malfunction are common in developed countries, the commonest diseases in the world are malaria and diseases that cause dysentery in children. And we're doing very little about that. Actually, good old Bill Gates, he's got a bit of money into those programs.
What do you think about the future? What's after genetic diseases for the West?
I'm not going to make any predictions.
Thank you. Now, Dennis,
Well, I wanted just to make a brief point. I just Googled ethics and brief learning in KAIS, and it comes up straight away. So anyone who needs that paper can download it off the web, and I've asked Zoe to print some off for after lunch. Can I just make one very brief point about pre-implantation diagnosis? Because some examples given, you know, I think sometimes people think of preventing Down syndrome or preventing people with disabilities. It's good to remember, I think, that a lot of genetic diseases affect kids, children, who, if they're homozygotes, are going to die painfully and slowly before the age of 7 or 8. Okay.
And sometimes within a few months.
And sometimes within a few months. But I'm thinking particularly of things like the lysosomal storage diseases, which are particularly cruel because the baby looks perfect baby when the baby is born, and you don't know anything is wrong at all. And then you see, you know, the slow downward course. And, you know, I've been, in my human genetics days, involved in families where they have one after another after another. So you have a mother who's gone through that 4 times just because of bad luck, you know. And I think it makes you want to believe in pre-implantation diagnosis, particularly, I think, for those cases where they can be prevented. I just want to put that side into the
well, that
discussion.
kind of reinforced the point I was gently making, asked that question. You know, who's going to say they shouldn't do it?
Yes, I would say so.
Absolutely.
yes.
Hello there.
I'm troubled by your response to Ernie's question. It sounds like you are in the school, the deterministic school.
Absolutely not. Absolutely not.
Okay, let me finish. That's why I'm confused. It sounds like sometimes you are and sometimes you aren't. Isn't it true, even in the case of Huntington's and hemophilia, that the effects of the RNA and the amino acid development and the protein development means that it's not a single cell, single disease, because there are other factors that are involved. And the most recent people are talking about systemic biology, where even the laws of chemistry and the laws of physics, the natural laws of chemistry and physics, affect the development of what happens in this process of embryonic formation.
Now you said, yes, sometimes I am, sometimes I'm not. That's how genes are.
Okay.
You know, we can, and this is the kind of dilemma of genes, that, you know, that they are, we can identify specific effects of genes, and I do mean specific effects, and yet we can also identify what you've referred to, we call these epigenetic effects, modifiers and so on, but nevertheless I will, somebody said, I will plant my flagpole that there are certain genes which, if you've got that mutation, you will get that illness. It may be modified, it may be less serious, it may come later, but you will have it. And that's not being a genetic reductionist. I'm not a reductionist, but nevertheless, as somebody who's worked with genes directly, I know that sometimes you can trace a direct pathway from the gene to the condition.
Thank you. Yes.
Cybrid cells, when you say you remove the genetic material, is that nuclear or nuclear and mitochondrial?
Oh, that's a very good question. We're talking about cloning here again. Cloning is done by this process called nuclear transfer. You take the egg, you remove the nucleus, which has got 99.9% of the genetic material in a mammalian cell, and you replace it with another nucleus. But it still retains these little energy organelles called mitochondria, which have got a handful of genes which are essential. And so you will have a cell that's driven by the nuclear genes, but where the mitochondrial genes, which are under control of the nuclear genes, are still present. And the interaction, shall we say, between a rabbit's mitochondrion and a human genome, a human nuclear genome, seems to be variable. You know, sometimes they seem compatible and sometimes they don't. You're quite right that the mitochondria remain. And so what people are doing, rather than taking the nucleus of the mammalian cell, they're fusing the whole cell with the empty egg. So you've now got human mitochondria in there as well. So it's a sort of belt and braces affair.
Well, I think we ought to end at that point, since it's taken us to 12:30. There will be, of course, the opportunity for questions for the panel, so do get that box filled up. I'm sure this is raised many more questions. So, thank you very much.